Hi all, I'm Shiwanka. I'm from Department of Chemistry and Biochemistry. Today I'm going to present one of my research projects titled as RAFT and ATRP Interchangeable Polymerization Methods. This project is a collaboration with Matyjaszewski Polymer Group at Carnegie Mellon University, and Julia is the co-first author of this project. So first of all, I will explain what is RAFT and what is ATRP. RAFT stands for reversible addition fragmentation chain transfer polymerization. So this is a one technique of polymerization, and it uses a chain transfer agent to control the polymerization. ATRP stands for atom transfer radical polymerization. It usually uses metal catalyst to control the polymerization. So both methods are controlled polymerization methods, which gives well-defined polymer structure. So this is the relevant mechanistic details for both techniques. So first equation is the main equilibrium step for the RAFT mechanism. So in this RAFT mechanism, we are using chain transfer agent, and this chain transfer agent is in a main equilibrium with the radical intermediate. You can see with it, which is in the red color, and in compared to that, in ATRP mechanism, we are using metal catalyst. So in here we are use copper as the metal catalyst. So this copper catalyst is in an equilibrium with two oxidation states, and it catalyzes the propagation of the polymer chains. But however, when there is a cross-linker present, a RAFT mechanism usually get interrupted due to the inability of finding monomers in a cross-link network system. This is a schematic representation of the two techniques in the presence of cross-linker. So the first one, the top one, is the ATRP gelation. You can see that due to the small size of the copper metal catalyst, it can continue the polymerization even in the presence of cross-linker. But in RAFT polymerization technique, you can see in the bottom figure, in this RAFT polymerization, the propagation step get interrupted due to limited transfer. So this can result heterogeneities in the network of RAFT compared to the ATRP technique. So our hypothesis is that ATRP give well-defined polymer network than RAFT in the presence of cross-linker. In here, our aim is to compare two polymerization techniques for network synthesis. This is how we synthesize our materials. We use hydroxyethyl acrylate as the monomer and also AIBN as the radical source, and all other conditions was comparable for both polymerization techniques. So these are the materials we synthesized. We prepared polymers with three different chain lengths, 100, 200 and 500. So you can see we synthesize materials with different cross-linking amount as well, such as 3% and 5%. So let's talk about some results. These are the radiological data we observed. This tan delta is the ratio between loss modulus and storage modulus, which is obtained from the rheometer. In ATRP networks, you can see the graph in the left side. You can see it gives sharp peaks compared to the RAFT network, which is in the right side. So the broad peaks of RAFT networks prove that it have heterogeneous network compared to the ATRP network. Then we did the size exclusion chromatography for both materials. In order to perform the size exclusion chromatography analysis, first we should degrade the networks because it's already cross-linked network, and to do that we used DTT to cleave the disulfide bonds, and you can see in the image it's cleave the disulfide bond in the cross-linker. So these are the results we observed for networks in the size exclusion chromatography analysis. So for comparison, we use free radically polymerized samples as well, which is shown in green color curves. So in here, you can see in the first top two graphs shows shoulder peaks for RAFT materials, which is in red color. So it means that it has less homogeneous network, so that is why it gives shoulder peaks. Also in bottom two graphs, it shows that RAFT gives broad peaks. So because of that, it has higher dispersity value. So the reason for this is the heterogeneous network in the RAFT. In ATRP, which is in blue color, you can see all peaks have well-defined narrow peaks compared to RAFT, so which proves that it has well-defined and well-controlled network compared to the RAFT. So as a conclusion, our size exclusion chromatography data showed ATRP technique form networks better controlled than RAFT technique, and also our rheological analysis shows further supported that the results from the size exclusion chromatography, which finally conclude that ATRP gives more homogeneous networks than the RAFT polymerization technique. So this is the end of my presentation, and thank you for listening.